Hi, so I'm here with Kat Lutz from Jackson Labs. She's a senior director there and she works with the mouse pharmacology. Kat, thank you so much for coming today.
Oh, it's my pleasure. Happy to be here.
Thank you.
Mhm.
So, like your mission statement and what you do and who you are. So, will you introduce yourself and then introduce Jackson Labs?
Sure. So my name is Kat Lutz. I'm the senior director at the Jackson Laboratories. I work in the in vivo pharmacology and the mouse repository. The Jackson Laboratory is a nonprofit institution that was set up right here in Bar Harbor, Maine, back in the late 1920s, 1929, I think to be exact. I've worked here for almost 30 years now, and I started here as a research assistant, and then went back to graduate school, did my postdoc, and eventually came back to what I think is the most beautiful island in the Northeast. And so we're located right up in the state of Maine on an island, Mount Desert Island, surrounded by Acadia National Park. It's a fantastic place to live. It's a fantastic place to raise your kids, and there's every outdoor activity that you would want to do. But I think, you know, all of those things being said, I think what really keeps you and people like myself at an organization like JAX for as long as I've been here, you know, is really the mission of the lab, which is quite fantastic. And you know, we are here to use mouse models to help support and improve human health. And we take that mission exceptionally seriously. And as I just recently described, the mouse models that we have are available to the entire scientific community as freely available as we can possibly make them within the licensing restrictions that they were given to us with. But making those mouse models, investing in things like genetic engineering to help promote the ease of use of those mouse models, investing in the phenotyping of the mouse models. We have the Center for Biometrics Analysis, which is just a huge endeavor by the Jackson Laboratory to really standardize the phenotyping of the mouse models, you know, that we currently have. And then certainly the in vivo pharmacology is where we like to do all the drug testing with the mouse models that we have. We collaborate, we partner, and we even do fee-for-service work so that for folks who want to test their drug and just don't want to deal with aspects of intellectual property or research agreements, we have a lot of different opportunities. I will say also that the lab would be, I would be remiss if I didn't focus on the research faculty at JAX. They're just an amazing group of people. And if you want to do mouse genetics and mouse research, you know, the Jackson Laboratory in Bar Harbor, Maine is really the place to go.
And highly reputable, and you make a fantastic partner to patient advocacy groups and to pharma partners and to academics. When we were first starting this work to figure out where was the best place for us to make the mouse model for 1B, time and time again, Jackson came up and said, you need to work with Jackson, they're the experts. And they're really driven to making the mouse accessible to anyone who wants to work with it.
What we feel very strongly about in our mission is that not only do we make these mouse models, but we also collect them from all around the world, right? So if there's a, you know, a CMT investigator in Italy, you know, who has the next greatest mouse model, we're going to contact him or her and say, you know, how about, you know, donating that mouse to JAX? And so when the mice come to JAX, we make them as freely available as we possibly can in terms of unrestrictedness. So you don't have to collaborate with us, you don't have to publish with us, you don't have to do anything except call our customer service representative and order the mice, right? And so we feel very strongly that having these resources, these tools, these little experimental models for these diseases we feel very strongly that having them widely available is going to be probably the single most important thing that's going to accelerate the development of therapeutics.
So tell me a little bit about the work at Jackson Labs and what you're doing and why it matters to drug development.
Sure, I'd be happy to. It's a little bit complex, but I think I can try to simplify it if possible. So a lot of times when companies are working in drug development, they want some indication of safety, but they also want indications of efficacy. So drug development companies are really there to sort of de-risk their products, the drugs that they may bring into the clinic. And in order to do that, they do look to a number of different model systems to try to get indications of whether or not their drug is either safe or efficacious or have target engagement, to the receptors or to the areas that they think that they had planned for. So where the mouse models come in is that they're an in vivo, they're a mammal, they're a system that really sort of is a little avatar, if you will, for people. So if we had a mouse model that mimics a particular human disease, you can often ask, did that drug ameliorate the symptoms of that disease in that mouse model? And that gives drug companies a certain amount of confidence moving forward with their drug.
And how important is the mouse study to the FDA approval process and showing that data?
Yeah, so certainly for safety, the FDA is very much concerned first and foremost with safety. And so a lot of the toxicology or the tolerability studies will happen in various animal models. Those may be in mice or they may be in rats, to make sure that the drug itself isn't providing any adverse effects, any off-target effects. And then what the FDA will also look for is some degree of efficacy. So do you have an idea about the therapeutic itself? Do you have some idea in an animal model if there has been any demonstrated efficacy, within that drug? Do you have information about the timing of efficacy, the therapeutic window?
And so mice are mammals and mice are teeny tiny mammals, but mice aren't naturally born with CMT. So how do you, or at least I don't think they are,
Yeah.
how do you make a mouse that has CMT? And how do you know that it has the human version or mimics the human condition? How do you do all that?
But since genetic engineering has gotten, so much easier to do, we've been able to take all of the different genes that are identified, for example, in CMT, looking at the mutations, whether they're loss of function, gain of function, point mutations, missense, stop codons, and we can, using CRISPR-Cas9, we can genetically engineer those precise mutations into various mouse models and also on different genetic backgrounds. And so that gives us a lot more power, if you will, to have mouse models that represent, for example, not just one form of CMT, but all the different forms of CMT.
Yeah, so in the case of CMT1B, which is the project that we're doing with you, this is a mutation of the MPZ. And this particular mutation, there isn't a mouse model for. And so talk to me about the value of creating a mouse model with a specific mutation, what that can do for people with that mutation, but also what that can do for people with a different mutation, even if it's in a completely different gene.
Sure. And I think where the idea of mimicking particular individuals' mutations comes to is that we have a lot more ability with various therapeutic strategies. Certainly we're not there with CRISPR therapeutics, but I think that, doing some of the somatic cell genetic editing will be completely in our future. But if we can mimic the precise genetic mutation in the patients into the mouse, we can then get an idea of severity, right? This mutation is really severe. This is really maybe quite mild, and this one, maybe relatively silent in mice, but for some reason, it has a very deleterious effect in the humans.
Mm-hmm, mm-hmm. And so, how do you see a pretty direct correlation between the way that mice present and the way that humans present? Usually, often, sometimes.
I'm going to say sometimes. We have had situations where the mice, I mean, they're models to begin with, so by definition, there's no identical mouse model. But certainly various mutations in various genes will probably will definitely give us a signature that looks very much like the human disease. In some cases, we have to do a lot more manipulation. Maybe we have to overexpress that particular mutation. And certainly in diseases of aging, it's a little bit more difficult to accelerate that aging process in the mouse, such that for diseases that most people wouldn't necessarily see until they were 50 or 60, putting those in a 4-week-old mouse can be quite challenging. So it does sort of run the gamut of, yeah, every single, phenotype that you see or clinical presentation that you see in the patient is manifested in the mouse, and then sometimes very subtly, and then sometimes not at all.
Okay, all right. And then once you've established that the mouse has the genetic defect that you're looking for, and it has some of the behavior that we're looking for, maybe in CMT their grip strength is not as strong, or maybe their balance is off, or maybe they even have foot drop. Talk to me a little bit about what happens to that mouse once we say, boy, this is a really great model of, in this case, CMT1B. What happens then?
Yeah, so once we actually genetically engineer the mutation into the mouse model, as you alluded to, the aspect of phenotyping that mouse and really interrogating the presentation of the disease in the mouse is next. And sometimes that comes along right away. You can see possibly a visible tremor. Maybe the mice are smaller. Maybe they don't have as much locomotor activity. So there are visual things that you can sort of key in and hone in on. But then there are also, some behavioral capabilities that we have that allow us to interrogate that just a little bit more. So some of the equipment might be a gait analysis, so you can put mice on a treadmill.
Okay.
And you can really analyze so many different parameters of that mouse in terms of its stride length, its swing, its stance. Is it a little bit atax? Is it moving back and forth? So those are things that you might not be able to pick up with your eye, but certainly these subtleties are very sometimes unmasked in a very efficient way, so that we can really quantitate exactly what the deficit is. We can ask, well, does that deficit get worse over time? Does it stabilize? Other things that you alluded to were grip strength, whether or not the mouse will stand up on its hind quarters, because it may not have as much strength as a wild type mouse. And then we look very carefully at electrophysiology, and that's again something that you can't do with the naked eye, but we can stimulate the nerves and look for a response in the muscle with a lot of these particular mouse mutants, and especially for peripheral neuropathies, we're looking for nerve conduction, and those are some of the parameters that you would also measure in the clinic if you were looking for just to monitor the progression of a particular disease. If you were looking for a clinical trial and you wanted an efficacy outcome measure, it may be stabilization of nerve conduction velocity, or possibly improvement over time. But so those are sort of the correlations of the things that you look for in the mice, the tests that you can do in the mice, and how they relate to the human condition. I think that one of the things we like to do with patient organizations and foundations is to really have a good understanding about what they have covered really well, and then maybe what resources or areas that they really need to push in. And so when we work with patient foundations, and we have thankfully a lot of support from NIH in precision genetics and working with various groups. And so we're very thankful for their support and what that can do, for the patient communities. I think we do try to understand, where we can be most helpful. And a lot of times it's making the mouse and getting it out into the research community for sure. But we do have, as I mentioned, so much expertise in evaluating that mouse model, looking for clinical similarities with the patient community. And most of the time we can do it at a level, at a depth and breadth that really can't necessarily be done in a lot of academic institutions. We have very large vivariums, we have access to a lot of reproductive technology skills, and so we can often get up and running a lot quicker than your average university, maybe professor could.
One who wants to work with it.
Yeah.
And lowering all the barriers to drug development. So thank you so much for your partnership. It's been a pleasure and a joy to work with you and to learn a lot about the power of a mouse model, and just thank you.
Oh, you're welcome. And like I said, it's our pleasure. It's been a great experience to go from basic mouse genetics and genetic engineering to having a greater impact on the patient communities, being able to to really move from just a mouse model that has a peripheral neuropathy to actually testing specific drugs and having that translational effect that I think is more powerful today than it has been ever before.
Yeah, thanks for saying that. There have been some really nice wins in the neuromuscular space, and I'm looking forward to sharing wins with you as well and CMT.
Yeah, great.
Thank you.